Start by turning on the pH meter and connecting the probe. Rinse the probe with distilled water, then blot it dry gently with a Chem wipe. Place the probe in the pH 7 buffer first. Wait for the reading to stabilize, then press the calibrate or set button. Next, rinse and blot the probe, then place it in the pH 4 buffer. Once stable, press calibrate again. Your meter is now properly calibrated. Before testing, rinse and blot the probe again. Place it into your sample solution, making sure the tip is fully submerged. Wait for the reading to steady, then record the pH value displayed. If you're testing multiple samples, rinse and blot between each one to prevent contamination. When finished, rinse the probe, then store it in electrode storage solution, not distilled water. This keeps the probe's glass membrane hydrated and ready for next time. Let's talk about acids. Specifically, what makes an acid strong or weak, how that affects chemical equilibrium, and what we can do to control it using buffers. First, the difference between a strong acid and a weak acid comes down to how completely they dissociate in water. A strong acid, like hydrochloric acid, fully dissociates, meaning nearly every molecule breaks apart into hydrogen ions and its conjugate base. A weak acid, like acetic acid, only partially dissociates. In this case, only a fraction of molecules release hydrogen ions, while the rest stay intact. Because a weak acid doesn't fully break apart, its reaction in water is reversible. This creates a dynamic equilibrium, a balance between the acid molecules that remain undissociated and the ions that have to split apart. At equilibrium, the rate of dissociation equals the rate of recombination, so the concentrations of each component stay constant, but not necessarily equal. Strong acids don't establish this kind of equilibrium because they dissociate almost completely right away.
Now, because equilibrium systems are sensitive, changes in concentration or pH can shift the balance. This is known as Le Chatelier's principle. To minimize these shifts, we can take certain steps. For example, controlling the amount of acid or base added to a solution helps prevent large changes in hydrogen ion concentration. But one of the most effective ways to stabilize pH is by using a buffer. A buffer is a solution that resists sudden changes in pH when small amounts of acid or base are added. It usually contains a weak acid in its conjugate base or a weak base in its conjugate acid. Here's how it works. When extra acid is added, the conjugate base in the buffer reacts with the hydrogen ions, removing them from the solution. When extra base is added, the weak acid in the buffer donates hydrogen ions to neutralize the hydroxide ions. In both cases, the buffer keeps the pH relatively stable, protecting the system from drastic shifts that could otherwise disrupt reactions or biological processes. So to recap, strong acids dissociate completely, weak acids only partially. Weak acids establish an equilibrium in solution, and we can minimize unwanted pH changes by using buffers which stabilize the system through reversible chemical reactions. Understanding these relationships between acids, equilibrium, and buffers help us control chemical environments, whether in a lab, in industry, or inside living organisms.